Hi, my name is Brady Ferguson, and I'm an undergraduate at the University of Washington in the CDAC Summer Lab program here at the University of Chicago this summer. I'm delighted to introduce Dr. Maryellen Giger, the A.N. Pritzker Professor of Radiology, the Vice Chair of Radiology, and a committee member of medical physics here at the University of Chicago. She's also the former president of SPI, the International Society of Optics and Photonics, and the former president of the American Association of Physicists in Medicine. Dr. Giger is a recent recipient of a C3 AI Digital Transformation Institute research grant, which will continue her groundbreaking research work in computer vision and machine learning with breast, lung, and prostate cancer, to name a few, but apply these methods to chest scan images used in detecting cases of COVID-19. I had the privilege of volunteering with Dr. Giger in her lab as a research assistant last summer, and I'm very happy to continue to do so this summer via the CDAC program. I'm looking forward to everyone hearing more about her exciting and boundary-pushing research. Please join me in welcoming Dr. Giger to the Summer Lab Speaker Series.
Thank you for inviting me to talk to you all. I think this is an exciting field, and thank you for allowing Brady to work in our lab again and extend her knowledge base. Okay, so I've been given 15 minutes. I'm going to describe in general what my lab does, and specifically my new C3 AI grant. Now, can you hear me?
Yeah, sounds great.
Okay, good. It's very noisy here. Okay. Okay, with every talk, I have to give my grants and conflict of interest, and I have a lot of different grants going on. I also have a conflict of interest because over the decades, we have translated our computer vision work in order through companies to get to the public, and that is the policy of the University of Chicago to let you know about that. So first off, let's look at the history and goals of the lab. So the focus of my lab is to develop AI for medical imaging and to actually continue on it to get it into clinical practice. Most of the work we do, but not all, the focus is on images that are clinically and routinely obtained on the population. And basically, we want to ask questions about the relationship between features seen in the medical images and the biology of cancer, for example, and in order to detect, diagnose disease early and give the right patient the right treatment at the right time. In addition, we want to improve the efficiency and the workflow of medical imaging interpretation. And this is an interesting side point, is that a lot of AI in radiology might not be in interpretation, but in improving the efficiency of the workflow. And so for some reason, okay, there it goes. Okay, so some of you work in computer science, and big data, but may not work in medical imaging. So let's think about, what is medical image interpretation? Well, medical images are meaningless grayscale or color scale patterns unless they're viewed and analyzed by an intelligent observer. For the longest time, that was the radiologist, and then came computer-aided diagnosis, AI. And I still think in the immediate future, it will be a combination of the human and computer for most tasks. And what does the human eye-brain system do? Well, it finds and locates signals in an image. It may classify, characterize, diagnose the signal as disease or non-disease, and then do clinical decision management on the patient. First, I want to clarify some terms. So you might have heard of computer-aided diagnosis, and you might have heard of AI, artificial intelligence, and I am loosely using this term orthogonal. I know I have some mathematical folks here in the audience, but the reason being is I've heard too many people, and you might as well learn while you're young, and they're saying, well, CAD didn't work, AI is going to work. Well, they're two different things. CAD Well, I should say AI is the computer algorithm that we develop that exists in the system to do different types of tasks, and everything we do is task-based. Whereas CAD is how do we use it, and computer-aided diagnosis or detection is just one method. So AI now in the medical field is being looked at as an aid to radiologists, a secondary reader, or as a concurrent reader, as well as a primary reader, for example, with triage, and eventually autonomous AI. Now, with all of these, if you're looking at AI, you evaluate the algorithm as a standalone. How well does it do what you're developing it to do? But ultimately, you need to evaluate it in the way it will be used. And in that sense, we often evaluate the end user, in this case, the radiologist, working on their own and then using the AI. Now, when I mentioned the autonomous AI, I haven't even touched on whether this is where AI will help interpret an image or to also integrate it with other diagnostics, say a blood test, some other clinical tests. So a lot of work that I do is in breast cancer. There's a lot of new cases every year, deaths, and roughly, in addition to that, 50% of women have dense breasts. So besides mammograms, we have many MRIs and ultrasound. So this is where oversight error by the radiologist might happen, confusing misinterpretations. So we have focused a lot of our work on this, and I'm talking about this now because we're translating what we've learned here to COVID, but there's nothing off the shelf. Everything has to be customized. This is just to give you an idea. This is our 1994 prototype system for computer-aided detection. And note here, this is when we used film, it was digitized. And here the computer is pointing out a cancer as well as a false positive, and here's a false negative. So the guts of the computer are very complicated, but the output to the radiologist is very simple. And I put this here because I'm sure many of you are all hyped up on deep learning and all. This was actually what I believe is the first publication of the CNNs in medical imaging interpretation. This was back in '94, though AI started way before that. This was more for the CNNs, which we term shift invariant neural network. So there's a lot of work being done, and I highly recommend we learn from the past and also learn from these brilliant young people who will make our future. So besides detection, we're also using AI to help determine, is it a cancer or not? So on the left is a benign lesion, on the right is a cancer. In the middle, it's a little more difficult. That actually is a cancer. And when we do this, we look at both human engineered methods as well as deep learning methods. And what we do is we automatically segment the lesion. We then pull out various features that are intuitive to the radiologist often. These are merged by some classifier to yield a signature. And we call this a virtual biopsy. This method helps explain what the computer, why the computer is given an output. But we also look at methods for deep learning. Deep learning, we don't have to segment the lesion. We don't have to extract out features. We keep training it by giving it images. And I do want to point out one of the reasons that we look at both of these methods is think how a radiologist is trained. They are trained by a textbook, learning these are the characteristics of cancers, these are not. But they're also trained by a residency where they go and they look at many, many, many images to learn on their own which ones are cancer and which ones not. So we continue to go through both of these avenues. Okay. And then I put this up. I can talk for hours on this topic, but I'm only giving it 7 minutes here. The point I want to take here is this is the same clinical case. It can be processed multiple ways for the radiologist to view. This is a central slice. This is a post-contrast MRI, details of which I can go through later if you wish. This is a subtracted slice, and this is a MIP image. So different radiologists will choose how they want to look at it, and many of them prefer the MIP image. So we were curious, you know, you often think with deep learning that if you have infinite number of images, you can just feed it and get your output. But often we don't have that luxury of an infinite number of images. So it makes sense to process your image or to focus the attention of the deep network on what you're looking at. So here we're looking at the tumor. And what we found was the computer, the deep network, also prefers the MIP images, just like radiologists. So often in development, I say, think like a human. How does the human eye brain system attack this problem? So should a computer. And also in our lab, we aim to translate. We're not doing homework problems. We develop the methods. We come up with novel implementations, and we want to translate them to the public. And we've worked with the Polsky Center to do this. And in the lab, you know, we're an applied field. We approach various medical tests, detection, classification, various diseases, breast cancer, thyroid case, emphysema, and others. We look at various imaging modalities and across various imaging scales. Today I'm talking about macro, but actually the image analysis that Brady works on is micro. And we look at various novel developments as well as novel implementations of AI. Okay, so the rest of my minutes will be spent on the new C3 AI grant. That grant is termed Medical Imaging Domain Expert Machine Learning for Interrogation of COVID. So we're basically looking at how to take the knowledge that we built up over the decades and apply it to this new problem. Now, with the COVID-19 pandemic, there's a pressing important public health need for computational techniques to augment the interpretation of medical images. There is the need for improved surveillance and early detection if there's a resurgence of it, as well as in monitoring medical imaging data. There's a role in detecting, triaging, and differentiation diagnosis. Is this a regular pneumonia or is this COVID? And then also for patients who are deemed to have COVID, can we predict what treatments should they be on and how do we monitor their response? Thoracic imaging with COVID has not had much attention, but there is a need both for chest radiography and computed tomography to be re-examined for their role. But in doing so, a limitation for improved interpretation is partially due to the qualitative interpretation of the images. You can start seeing the parallel with breast image interpretation. So if we can go from qualitative interpretation to quantitative interpretation with AI, that could aid in the interrogation of these medical images. So these are the aims of our grants. We're now establishing a COVID and non-COVID thoracic imaging database. This has been moving along quite nicely. We're currently also developing machine intelligent methods. And this grant, by the way, was just funded in June. So we also will investigate the explainability or visualization of COVID AI. And we will also do, of course, statistical evaluation of them. This is a one-year grant, and then we were planning to submit to NIH. An important part of this research is our research team. It includes medical imaging domain experts in computational machine learning methods. So those are folks basically from my lab. It also includes medical imaging domain experts in the clinical interpretation of thoracic CTs and chest x-rays. Those are clinical radiologists. They do a very good job. We need to, we always collaborate and integrate them in the research. And we also have an Argonne expert who's an expert in applying high performance computing. So multidisciplinary teams are a must for this field. We're going to be building on our prior AI research in breast cancer images, as well as our research in thoracic imaging. We've looked at pneumothorax detection, coronary artery disease, as well as an emphysema assessment. This is a flow chart from a recent student who just graduated, applying AI along the entire workflow in first identifying the type of images and then making sure to correctly process them to find pneumothorax, and then using visualization techniques to see if where the computer is pointing is actually where the disease is. I have a commentary on explainability. When you think about it, if an AI system is very, very good, in the long run, people may not they won't ask, well, how did it do it, if they have that confidence? But we're not there yet. So it's very important that the AI is able to give output that helps explain why it pointed to a certain area as suspicious and not to another area. So keep that in mind as you do your work. One of the methods, this is being done by Jordan Furman in my lab, is he's done work on emphysema in lung CT, and now he's using that knowledge with a cascade deep transfer learning approach to identify in this first round which patients should go on steroid treatment and which ones should not. And he's gotten some very promising results, and throughout the year you can be updated. Again, this has only been out. I want to show this for explanatory AI. This is worrisome. These are heat maps in the detection of pneumothorax. And what you'll find is while all of these did say there was a pneumothorax in the image, only 33% of them correctly identified the location. So there's other things. And we were very careful not to use images with other items such as chest tubes and all, which caused that problem. So I think there's a lot of work to be done still in exploratory AI. So we aim to develop an early stage tool for surveillance and early detection, as well as use our methods for virtual biopsy to monitor those on treatment. We have 2 avenues going on in the lab, and we have both on CT and chest X-ray. So I'll end with this slide again. It's important that if you're going to do AI in medical imaging, to specify your clinical question. Make sure you have solid images and truth. I mean, there's databases all over, but you need to make sure that you have it right. And remember, garbage in, garbage on out. Think about what you're inputting to your algorithm, and could you pre-process it to help that. We are very big on robustness as well as methods to evaluate. And I'll end there for my 15 minutes. These are students, research lab staff, and collaborators. And I always, this is from, maybe 2 summers ago. I always take my lab pictures during the summer. Currently, I have 6 summer students, and it's called image enhancement to take the pictures with them. So thank you very much. Are there questions?
Thank you very much, Maryellen. I think we'll leave questions until after Rick, so we can have sort of a general discussion about data science, AI, and all things medical. So that was perfect timing. Thank you very much for that. So Rick will, actually, we've got someone, Arthi, I think, is going to introduce you today. So Arthi, would you like to do the introduction?
Yeah, sure. Hi, everyone. My name is Arthi, and I'm an incoming freshman at UChicago in the CDAC Summer Lab program. I'm excited to be introducing Dr. Rick Stevens, the A&L Associate Laboratory Director for Computing, Environment, and Life Sciences, and a Professor of Computer Science at UChicago. Dr. Stevens is a member of the American Association for the Advancement of Science and has received many national honors for his research, including an R&D 100 award. His research includes building innovative tools and techniques for biological science and infectious disease research, as well as approaches to advanced deep learning to accelerate cancer research. He also has extensive experience with high-performance computers and leads A&L's initiatives to deploy the first exascale computer in 2021. Today, we're going to hear Dr. Stevens speak about a multi-laboratory team of several hundred researchers and how they're using advanced AI and supercomputers to analyze the COVID-19 to find treatments, as well as predicting how it will spread amongst populations. Please join me in welcoming Dr. Stevens to the Summer Lab Speaker Series. Thank you.
Thanks, everybody. Can you guys hear me okay?
Yep, perfectly.
And can you see the slides?
Yep.
Okay, good. All right. So I'm gonna, and generally what my group works on, I have about a dozen or so PhD students and about 30 or so staff members that are part of my group, is the general concept of applying AI, where we, in the last decade or so, high performance computing in general to problems in science. That's kind of our space, and computer architecture and software in that space. So what I'm going to talk about today is what we've been doing on COVID-19 related work since late February. So I'd start by saying we need to understand the enemy here, and say a little bit about the virus and how we think about it. So some of you probably have read or seen presentations about the virus. So this is the SARS-CoV-2 virus is a coronavirus. It's a single-stranded RNA virus, which means that it codes, its genome is coded as an RNA molecule in the center. It's a particle about 100 nanometers in diameter. And it is only transmitted, it can only replicate inside host cells. It's not like a bacterium, which has a life of its own, and viruses are actually not at all alive. And it's very similar to the SARS-CoV-1 virus, about 80% similar to that. It's about 50% similar to the influenza virus, and a little bit slightly more similar to the MERS virus that emerged 10 years ago or so. To attack this virus, and I'm managing my dog here who wants to bark because the UPS driver likes to ring the doorbell when they deliver a package. So she's under my table here. Fortunately, or unfortunately, because the community had been working on SARS-1 since over a decade, a lot of the protein structures that the virus genome codes for, we actually had structures for. Here's a picture that shows the proteins that are coded for in that genome, for ones that we had structures for. And the purple coloring, if you can see that, is where in these protein sequences there are mutations, basically the differences between SARS-1 and SARS-2. And you can see that some of the proteins are almost identical, 99% plus percent similar. Some have a fair amount of variation, like the spike protein that's on the outside of the virus. Now, to understand this thing, this is in a class of viruses that have very similar behavior in terms of how their genome is encoded and how they work. It's basically 30 kilobases of RNA, and it codes for 3 big regions. Viruses tend to do things, code for things called polyproteins, which are a whole set of reading frames that get translated at once, and then the protein sequence from that gets processed into the functional elements that actually carry out the work. And about 2/3 of the genome is used for proteins that actually facilitate replication of the protein. So all the proteins here kind of on the left are these things called NSP 1 through 16, are proteins that code for things that take over the machinery in the host cell and let the virus replicate. On the right-hand side, there's about 7 or 8 proteins that are actually code for things that make the viral coat and are called structural proteins. All the pictures on here are cartoons of the protein structures, and these were all solved at the Advanced Photon Source at Argonne over the years. And we solved many versions of these structures, sometimes with drugs bound in them, so we can understand how those drugs work. So the high-level challenge that we're working on is trying to find drugs that bind to these proteins that will gum up the works. Think of it as throwing a wrench in the gears, and you're trying to stop the virus from doing what it's trying to do. And what this looks like computationally, the first order computational task is what we call virtual screening. So we build a 3D model of the protein, and we identify where in that protein is the active site. And then we do a process of fitting, or sometimes called docking, of small molecules and and try to get them into that binding site and measure computationally, estimate the binding energy. That's how well they actually bind to that site. So that's the first order task that we're trying to do. And you can see cartoons here. This is one of the proteins, NSP16. Its function is to basically cap the RNA as it's getting ready to go in the virus to exit. And this blue region here is a subset of that protein surface where is the active site and is where we're trying to get the drug molecule to stick. And we screen both existing drugs. So on the right is an example of a known kinase inhibitor that does bind to that site, and it has a estimated free energy of about -14 kcals. It's a pretty good binder, but on the left is a result, a best score out of 6 million new molecules that we looked at that has a better score. So this is the kind of space that we're in, which is models of each of these proteins and then millions of compounds that we're trying to dock. And the challenge is trying to keep track of all of this stuff and doing it in a high-speed fashion. There's over 10 to the 60th possible drug molecules, that is small molecules that are drug-like. And of course, even if we had the world's, all the machines in the possible universe, we still couldn't search that. So we can't search all the possible drug, hypothetical drugs. In terms of an actual database, we have a database of about 4.2 billion molecules that people have actually put some energy into creating conceptually or physically. And one of the challenges is, can we screen all of these targets? And the way to look at this table, these are all the different protein targets. On the left are the green stars are the things we actually have structures for. Things on the right that are the blue stars are things that are plausible drug targets, that is the molecules. The protein's something we understand. It's a function we understand. If we interrupt it, it's likely to disrupt the virus life cycle. And it's something that whose function we know, and we have a reasonable structure and so forth. So there's about a dozen or so targets on this list, and we want to screen those dozen targets on these 4 billion molecules. And that task, if you just tried to do it naively, is still not doable on the world's fastest machines. And so we have to be more clever about that. On the right-hand side here, this is a cartoon of a cell and a cartoon of the life cycle. And highlight here, I highlight kind of 3 regions. There's part of the life cycle where the virus is getting into the cell. And so disrupting viral entry is really an important strategy because that prevents infection at the very beginning. If you get the virus into the cell, the virus comes out of the RNA comes out of the capsule and it starts to take over the replication machinery. In the middle area here is the replication and processing a center or complex is called, and there's a dozen or so targets that we can target inside that for disrupting the replication part. And then on the bottom is processing that the host does to react and try to create, the host will try to fight the virus. The virus, some of the secondary functions of these viral proteins is to disrupt those host functions to create an environment that's more hospitable to it. And the virus also has to exit the cell, and we can potentially block that. So we have these kind of 3 kind of zones where we're trying to block, and we have about a dozen or so targets and about 4 billion molecules. It also turns out that these viral proteins interact with the host proteins. In fact, maybe hundreds of host proteins. So over evolution, I mean, this virus' natural habitat is in a bat. It came into humans probably through some intermediate host that we don't understand, but it co-evolved with bats for millions of years. And so, most mammals have these kind of viruses. So in the bat physiology and the bat microbiology is very, or not microbiology, but the cell biology is similar to that in humans because we're mammals. And so it evolved this notion of interacting with many, many different molecular pathways, including modulating the immune system and other things, and trying to tinker or tear that apart into understanding these pathway interactions gives us additional drug targets in the host, and maybe on the order of 50 or so good drug targets people are working on. Now, if you think about this space, it's like a giant funnel, right? At the top, we've got something like 4 billion molecules that we know about, and maybe another 100 billion or something that we could relatively easily make if we knew what to make. And we want to get it down to a small number of compounds that we can actually make and then test in a experimental assay to move on towards drug statement. So it's basically this funneling process is the computational challenge. Up in the right here is an example of one of our AI-based systems that's generating molecules kind of in response to the binding energy in a docking system. In order to drive this with AI, we had to gather a large amount of data. So there's a team here, Kyle's on this team, and a bunch of folks at the university and our group at the lab. So we spent a couple of months actually gathering and generating on the computer ways of representing these drugs and representing the protein such that we could use deep learning methods to actually help us accelerate that screening process. And that produced about 60 terabytes of data. So this is not something you could do on your laptop at home. It requires actually very large infrastructure. It includes computing things like molecular fingerprints, which is a graph representation of the molecule, descriptors, which are numerical values that characterize the molecule, images. Maryellen was talking about images and about attention models, where we can understand how the network is actually focusing on images to make decisions. It turns out one of the best ways to build an AI system that could predict the output of a docking system is to generate images of the molecule, just like a chemist would look at the image and try to have some intuition about how that thing would work. The neural network wants to do the same thing. And so it turns out image-based models actually work incredibly well for doing this. The overall pipeline looks something like the following. There's a fast part, which we can use machine learning methods to search through millions and billions of molecules once we have trained model. And we can predict properties, binding score, toxicity, and so on. And there's a process at the bottom where if we need more data, we have to then launch computation, molecular dynamics computation, or experimental lab work, and that takes a long time. So we're trying to focus on accelerating the top part that allows us to search the large databases as fast as possible. Ultimately, where we're going with this is we want to automate the wet lab parts. So we'd like to get this drug development cycle to work essentially with humans only in the supervisory role. So the screening is all done by the machine, a combination of simulation and AI, and the synthesis would be done by automated labs after the machine tells the system what to make, and the system would make the drugs, take those drugs, and then screen them on, say, whole cell assays or functional assays, again, all automated. That's where we're headed with this, that we won't get there in a year, but in the long term, this is where I think this kind of work is going. One of the things we do in the very short term is to take existing drugs and make sure we screen all existing drugs, because the fastest way to a therapy right now is to repurpose an existing drug. And we've done this for the 4,000 or so drugs that are licensed around the world. And this just gives you some sense of the kinds of hits we get against different targets. These first column here is the main protease. The other protease, it splits up the polyprotein when it's in the replication cycle, and a couple other functions there. And then this one function on the right-hand side, which is a host protein that's required for the virus to get into the host. And you can see these drug names here. These are all existing drugs that scored high in our scores, and these are ones that we're actually testing in the lab, as well as compounds that are not yet drugs. Once we have some of these hits, we also want to understand the dynamics. And what I can show you here, I won't go into this, I don't have time, but different drugs will bind to a protein and they will stabilize or disrupt the structure of that protein. And trying to understand how the drug actually alters the shape of the protein, or the conformation of the protein, and how it alters the protein function is also something that we want to understand. And whether or not a protein naturally has more than one state, and whether the drug is effective in all of those states or only in one of the states, as indicated by this clustering diagram. So everything we do is in the open. We produce a lot of software in our group. We scale it up on the large scale computers. I mentioned the large scale data and all the models, and it's a one big kind of happy family. We're using about 10 different supercomputers all around the world. We're using the supercomputers at Argonne, the supercomputers at Oak Ridge, the supercomputers down in Texas and San Diego, Pittsburgh, Germany, and other places. And as as Kyle mentioned, I mean, or maybe Artie mentioned, when we started this effort to pivot, we normally work on cancer and bacterial infectious disease. We pivoted towards COVID the end of February, early March. And within a couple of weeks, we had over 200 people helping us as part of this, including the biosafety level 3 lab that the University of Chicago runs at Argonne, which is doing host cell screening on live virus, as well as collaborators kind of all around the world. So I'll stop there. Happy to answer questions when we get into the discussion part. Thanks.
Awesome. Thanks a lot, Rick. This is the best timing I think we've ever had in the several years of running summer seminar series. So thank you both for being so efficient with your presentations. So I'll turn it over now to students. Please feel free to yell out with any questions you might have. No question is stupid in this context, so please feel free to ask anything you were thinking about as we went through those two talks. Obviously, there are a lot of similarities between the talks, but also some differences as well in terms of the approaches being applied. Again, I have no prizes, but I can guarantee you Katie will give you a prize for the first question asker.
We have an unofficial 10 points to Gryffindor system for folks. So if you have a question, feel free to unmute and go for it.
Okay, I have a question. This question's for Rick. My name is Max. I'm in Ian's group, by the way.
Hi, Max.
So these drugs, they interfere with docking, binding to the virus, right, to the virus protein. Is there any parallel study of the effects of this drug on other human systems, other sites where it could bind and potentially interfere with pathways that are essential to our survival?
Yeah, absolutely. So in the normal drug development cycle, as you develop leads, these are, when we do docking, they're called hits. It just means they look interesting. To move from a hit to a lead, you have to actually start asking and answering a lot of questions about the molecule. Is it toxic? Does it have off-target effects? Does it bind to other proteins in the host that might cause a problem? Is the dosage it need for effect so high that the therapeutic window is too small? Is it soluble? Would it have to be injected versus taken orally? And all these things, right? So these off-target effects, there's actually a way to use the same technology that we're using to screen for those. And so it's called a counter screen. So you would use systems biology network analysis to identify targets that might be in the same pathways, and you would screen those. And what you'd want from an ideal outcome is to have low binding affinity to those targets and high binding affinity to the target that you're aiming at. So the counter screen is for showing that it doesn't bind to those targets. And then you would go to tox studies, which is mainly looking at whether it passes through membranes in the liver, liver metabolism, and so on. So there's a variety of things besides just this front end that we have to do, but the same methods can apply.
Do we have a good, as a body of knowledge, as a group of scientists, or as a consensus, do we have a good catalog of these off-target binding sites that it could possibly have high affinity with?
Yes and no. So the human proteome, we don't really know how big it is, but it's big. It's probably 30 or 40,000 proteins, and of course complexes and interfaces and complexes. So it could be 100,000 potential binding sites that you'd want to, if you were in a theoretical sense, trying to show that it doesn't bind anything else. Practically, we don't have structures for all of those, and even if you had it docking on 100,000 targets, would take years. So you can't actually do that. I think what we do is we try to find pathways where, because of the molecular shape and the function that it's inhibiting and its similarity to a natural, say, substrate or ligand, if it's a metabolite, look for other systems in the body that operate on those similar molecules, and then look at those places first. Because that's the most likely place where it's going to have an off-target effect. And for many drug classes, we can do that.
Good answer. Thank you very much.
Thank you, Max, for leading the way. And obviously, Max is not eligible for the prize that I just promised. So
what the hell, man?
We now need one of the summer students to ask a question. Does anyone have one that they'd like to ask? Obviously, the bio side of things is well over my head, and I'm sure over over many people's heads, but there's obviously computer questions here as well that might be relevant
There's
to you.
Question in the chat here.
Oh, great.
Yeah, I can read this out. So this is a question for both Maryellen and Rick. So as a student, this is from Yuja. So as a student coming from a computer science background, how much medical knowledge would one need to catch up to do research related to medical imaging or health informatics? Or how can CS students better leverage their strength within these fields of medical imaging?
Well, I could take a stab at that. Many of the graduate students in our program come from physics, so they don't come from a medical background. They come in through our medical physics program. Many of them have a lot of computer science background also. It's more of being a creative, intelligent, analytical person who's willing to learn. The medical imaging, the medical part, you learn along the way. Our teams are multidisciplinary. In my lab, all the students have a clinician on their dissertation committee. So I would say learn how to be a problem solver. Now, my lab, which is different than the computer science department here at U of C, is more of an applied. You know, we use methods of AI and customize them. We customize what goes in them and how we use the output in an efficient way, and how do we link that up with how a human might use them. But to me, if you're taking courses, I don't know, is there a course on problem solving? Hopefully you're in that field. So it's most of it you will learn along the way.
Yeah, I would second that. I would say that the way I think about it, I don't believe the world actually, I don't think disciplines really don't exist. Those are a figment of some medieval university architect, right? What really matters is people who are enthusiastic about problems, they're hungry, they're not afraid, they're fearless. They're not afraid to jump into something where they don't understand all the details initially. Now, of course, they might have to understand a lot more than details eventually, you know, after they get going, but you're going to have to teach yourself new stuff all the time. It doesn't matter whether you're starting from computer science or physics or biology or, you know, a clinical track. To be operating in this collective space, you're either going to have to learn. If you come from the biology side, you're going to have to learn a bunch of math and machine learning and computer science and coding. And if you're coming from the computer science space, you need to get your head around, you know, a protein or a pathway and a tumor or, you know, or imaging or whatever. But that shouldn't be viewed as a barrier. It should be viewed as an opportunity to kind of, you know, learn new things. And we try, I think our group is very similar. We have chemists, molecular biologists, physicists, chemical engineers, computer scientists, mathematicians, statisticians, all these random people. And we just pass it's like the problem's a hot potato and you pass it around. What do you think of this? And we go around the circle. And getting enthusiasm is more important than knowledge. I'll use that Einsteinian quote, I guess. You can learn the knowledge. It's hard to teach people how to be enthusiastic about a problem. And that's what we're looking for, is that thing that caused you to wake up at 2 in the morning and sit down in front of your machine because you had an idea and you can't wait till the morning to work on it.
Awesome. That's a great answer. Thank you for the question as well. Do we have any other questions? I'm led to believe that Arthi might have a question.
Yeah, I have a question. I was wondering if you had any books that you recommend that like touch on computational science or like the intersection of like medicine and computational science.
Rick's got a whole bookcase behind him, I see there.
That's a picture of my office at the lab because I'm at home.
Oh, is it?
Yeah, you can see if I move around.
Yeah, I knew it was a fake background. I just didn't know it was
it's my office at the lab. Not that I really miss it that much, but you know, my personal take is that books on this topic are not so useful. There's review articles, right, on specific problems. I think those are useful. I think, of course, watching people lecture on it's useful. I mean, the problem with I mean, this is just a problem, right? By the time a book gets through the whole publication process, it's usually several years out of date. And often people that have the time to write books aren't actually on the leading edge anyway. And so I'm not dissing all books. I love books, but if you want to learn about computational, if you want to learn about something that's in this interdisciplinary space, it's hard to learn about it from books. It's probably easier to listen to a bunch of lectures. When university gets back up and running in the fall, we have this beautiful computational in science series that's put on by physics that hosts it, but it's always ranged. Go to these talks. At first, it won't make any sense to you, but after a little while, you'll start remembering things. It'll start to make sense. Of course, if you want review papers, I can point you at things to read. Maybe Marianne has a completely different theory about this, but I mean, that's my take on it.
No, I totally agree with Rick. And if you want some of where AI, computer vision is in medical image analysis, whether it's in detection, diagnosis, therapy, drop me an email and I'll send you off. Or maybe I could send some to Katie and she could put them wherever you put them.
I mean, this topic of combining AI and computational science is really new. I mean, traditionally AI people have worked on problems that humans can do that we don't have algorithms for, right? Computer vision, speech, natural language, things like that. And the systems weren't particularly capable, and so they didn't really cross over much in applications in science. Like, for example, if you wanted to do AI for classification, applying it to a problem like, say, galaxy structure, well, that's become hot in the last 10 years. But prior to that, it was not really possible because the computer vision algorithms were pretty bad. And the training data didn't exist and so on. So you have this kind of strange situation where the AI methods are starting to work quite well in various areas, and they're starting to become good enough that we can apply them in science and, of course, medicine for sure. And a lot of it's open territory, right? One thing we're particularly interested in is this idea of using AI for designing things, right? Designing molecules, designing materials, designing proteins, designing organisms, right? And this is a very different use of AI because it's not just simply replicating what humans can do, because humans can't do that very well. They can do a little bit of it. They think about it a lot and they try. But what we're interested in, many applications in science aren't just kind of replacing something that humans are pretty good at with a machine to automate that, but actually to go beyond what humans are capable of doing. And that's actually a very open space right now.
So we've got some great sound bites today. Everyone, don't read books. I think that's one we'll take. Next question.
Buy books.
Yeah, yeah, exactly.
Read them.
Questions from the crowd. It's easier when I'm in person. I can start staring at people. Here I can't stare at them and make them feel bad about it. I guess it's not appropriate to call people out. So
I'm happy to like fill the void with a question that hopefully will encourage other student questions. This is a selfish one that I'm interested in hearing both of your opinions on. But to say that, COVID is an urgent and timely enemy, as Rick said, is obvious. And you both have kind of storied careers built on work with other viruses, work in other fields. What was the pivot towards working with COVID like? And what would you say is the most kind of like unique challenge that COVID itself poses compared to other enemies or problems that you've approached before?
Well, part of the reason for pivoting was because it occurred. And the issue of images are a very good way to see what's going on inside the body. In fact, in some cases, they're using images to see lung involvement even without other clinical signs, some of the asymptomatic folks. And so while nothing is off the shelf, nothing is, oh, it works on breast cancer, so we can just take it and use it on COVID. But it was there. We felt that we should get involved. We have a lot of resources, both in past knowledge and others. And we do customize what we've done in the past for other disease sites. I think the challenge with COVID was, well, and people say that a lot of publications are coming out. A lot of them are poor, and they're getting better. And a lot of people just rode the wave of it. But it's getting a very solid data set that is correctly curated. Get a data set is a data set. Anyone can get a data set, but a really good one takes a lot of effort, and that's what we're working on now. But a lot of the tricks of the trade that we had from other diseases we can apply, and we know when something is working, how to look for the red flags, and we hope that by doing that, imaging for COVID can become routine, so that if one we've been also working in lung cancer screening, low-dose CTs, where someone they used to call it lung cancer screening. Now they call it lung screening, because when you get a low-dose CT scan of the lung, not only do you see cancer, but you can assess emphysema, osteoporosis, coronary artery disease. Why not be able to throw in now COVID in the future? So while I only had 15 minutes, so I talked on the breast cancer. We also have a major collaboration on the lung cancer, low-dose lung cancer screening. And now we could also put in analysis for COVID. So all the puzzle pieces fit.
Yeah, I can
long answer to a short question.
No, that's good. I think from our group, I mean, we've been working in bioinformatics and infectious disease, comparative analysis, and therapeutic design essentially for 20 years. And of course, in bacterial infectious disease, we have a huge challenge right now with antibiotic resistance. And so for the last few years, we've been spending a lot of time on that problem, which is a very interesting problem, because there's a arms race between the bacterial populations and the chemists and also policy people about how to use antibiotics and how to devise new strategies that will give us a good armament of tools for fighting bacterial infectious disease. And virals are really hard. I mean, in the last 50 years, there's only been about 90 licensed antiviral drugs, and in the HIV case, it took about 20 years before there was actually a good regimen. And Hep C, it was a little bit faster maybe, but viruses are really hard. And unlike bacteria, you're really, you can kill the bacteria independently of trying to understand how the bacteria is dealing with the host. Whereas in the virus, it's really this complex interaction that you have to disrupt in a way. And protease design is really hard. So, I mean, it's really challenging. Of course, it's urgent. And there's also a huge push, I would say top-down push from policy people saying, well, we have all this machinery and these supercomputers and all this labs that we built after 9/11 for biothreats. How can we use all that stuff to actually make some headway on this problem? And so we were kind of in the right place at the right time for that. And people are enthusiastic. And if you're going to work on something from 6 in the morning to midnight every day, you might as well work on COVID right now, because everything else is harder. But cancer is also one that's there, and we've been working on cancer for a long time as well. And one thing that we're starting to think about is the country needs the ability to actually react to these things more appropriately. It can't just be like the scientific community bouncing around like a ping pong ball from one crisis to the other. There needs to be kind of a whole infrastructure around doing this. These little grant opportunities that popped up, for COVID, that's great because it gives you some free energy, but it's not sustainable. I mean, you can't build a whole lab on 1-year grants, right, and stuff like that. So there's this kind of need right now to really rethink what's our preparedness strategy need to look like from a scientific standpoint should we have standing teams that work on things, like from my standpoint, on like orphan diseases, so that we're constantly designing new drugs and tinkering and have a whole pipeline, so that if you have a new threat like the new swine flu that might become the follow-on pandemic, then you're actually kind of in a warm start. You can just kind of slightly pivot and you're already to go. And I think that's kind of where we think things need to go.
Yeah, that's a really good point, Rick. And I guess both of you are great examples of this ability to sort of pivot. If there was more steady state able to explore these things, then it would be much more responsive. So I guess we're coming to the end of the hour, so we'll finish with one final question. So I guess a question here that's probably relevant to everyone in the room. If you guys can think back to when you were at a similar stage of your career, late high school, early undergrads, what would be the one piece of advice that you would give to yourself around? I know that's a tough question, but what advice would you give to yourself at that stage, knowing what you know now from your several decades, in Rick's case at least, of experience?
Well, it's going to sound really simplistic. I mean, I've always was and always will continue to go after interesting opportunities, and I work really hard and enjoy working with people. But I think I would tell myself not to sweat the small stuff. I'm enjoying the same stuff I did years ago much more now, because it's don't worry about too much. Just let it happen. Sounds a little crazy, but.
Rick, you're muted.
Siri muted me. I think very similarly about this. When I was exiting high school, of course, I was primarily thinking about physics and math, and I was fascinated with computers, but computers were kind of a distraction from physics and math. But in hindsight, of course, my whole career has basically been around computers, and then biology, because I was always interested in biology and evolution. But your path is going to be very winding, probably. It's not going to be a straight line, and you should not worry about the fact that it's not going to be a straight line. If you get interested in something, go after it, and don't worry about what other people think. Skip classes and go into classes that you actually like. I mean, when I was studying, math, occasionally I'd have a bad professor, so I'd spend most of my time hiding out in an evolutionary biology class instead of going to my math classes, because just because I was more interesting, and that turned out a good decision. Maybe it was locally bad. I probably got a crappy grade in it, but grades don't matter either, right? Grade doesn't matter. It's what you actually get out of it and what you decide to do with it that matters. And your brain is kind of sponge-like, right? And so what I find is the fact that early on I spent a lot of time studying different things. It comes back now and is useful, because you can kind of say, there was this, and there was an example, one of my PhD students a few years ago, we were working on a problem, and it turns out that we were trying to understand horizontal gene transfer. And I remembered a parasite coevolution in biogeography theory. Okay, so evolution, biogeography is when you have islands form and little populations get separated, and so they evolve kind of independently. It turns out that people had studied how mites and the mite host had co-evolved in these biogeography situations, and how you could actually align the phylogenetic trees of the mites with the phylogenetic tree of the host, and they would align. And you could actually look at the interior branches, and you could see when islands had separated and so forth, right? So you could actually read geology from the phylogenetic tree. And you say, okay, what the hell does that mean? Well, it turns out that we did the same thing in studying horizontal gene transfer, where genes don't pass in a Darwinian sense from the parent to the child, or bacteria from the mother cell, but instead are transferred using some other mechanism, natural competence, or whatever. And it turns out you can take the evolutionary tree of the individual proteins and the evolutionary tree of the organism, you can align them in the same mathematics that allow you to describe host-parasite relationships, and mapping it onto geology would allow you to reconstruct the horizontal gene transfer pattern in bacterial evolution, okay? So I wouldn't have really and that turned out to be a thesis problem for him, from one of my students, because we made that connection, right? And so don't be rigid in your thinking, be flexible, be open, have strong opinions, but make them weakly held. So if there's new ideas, be willing to abandon some bad idea and glom onto the new one, but then defend the new one like crazy, right? Until the next better idea comes along. And that's the best way to proceed, I think.